Step outside at 7 in the morning on a clear day and look toward the eastern horizon without staring directly into the solar disk. You feel the cool morning air against your face, but something far more consequential is occurring at the subatomic scale across the surface of your eyes. Particles of light that were generated in the thermonuclear core of the Sun hundreds of thousands of years ago and which traveled 93 million miles across the vacuum of space in precisely 8.3 minutes, are now entering your visual system at 300,000 kilometers per second. Within 90 seconds of standing in that ambient morning light, hundreds of trillions of photons pass cleanly through your tear film, penetrate the clear avascular tissue of your cornea, transit the anterior chamber, traverse the crystalline lens, and project across the vitreous cavity to strike the multi-layered neural fabric of your retina. Your retina is not merely a piece of sensory tissue, it is an outcropping of the central nervous system, an exposed piece of your brain wired directly to the outside world. When these photons strike the back of your eye, the vast majority of people assume they are simply being captured by the classical photoreceptors, the 90 million rods and 4.5 million cones that construct your visual perception of the sky, the silhouettes of trees, and the asphalt beneath your feet. But nature has engineered a completely independent sensory pathway that operates in parallel, blind to conscious imagery, yet utterly dominant over your internal biology. Buried within the innermost ganglion cell layer of the retina, sitting forward of the rods and cones, lies an elite population of specialized neurons known to neurobiologists as intrinsically photosensitive retinal ganglion cells, or iPRGCs. Comprising only about 1 to 2% of all retinal ganglion cells, these extraordinary neural transducers do not require synaptic input from rods or cones to sense the external world. Embedded within their plasma membranes is a specialized photopigment called melanopsin, an opsin protein coupled to an 11-cis retinal chromophore. Melanopsin is tuned with exquisite evolutionary precision to absorb photons vibrating within a very narrow spectral band, the blue-cyan range spanning 460 to 490 nanometers. The morning sky is rich in precisely this wavelength. When a photon within this exact spectral window strikes a melanopsin molecule, it triggers a photochemical cis-to-trans isomerization, activating a G-protein-coupled signaling cascade through the GQ/11 subfamily. This activation stimulates phospholipase C, hydrolyzing phosphatidylinositol 45-bisphosphate and triggering the gating of transient receptor potential channels. The result is an immediate, sustained inward flux of extracellular sodium and calcium ions, depolarizing the cell membrane and initiating an unbroken high-frequency train of electrical action potentials. These action potentials do not travel along the traditional optic pathways to the lateral geniculate nucleus or the primary visual cortex at the back of your skull. You cannot consciously see this signal. Instead, these electrical pulses are diverted onto a dedicated private neural superhighway known as the retino-hypothalamic tract. This monosynaptic projection travels directly into the anterior hypothalamus, terminating inside a bilateral pairing of microscopic structures positioned immediately above the optic chiasm, the suprachiasmatic nucleus. Comprising a tightly packed cluster of approximately 20,000 specialized pacemaker neurons, this tiny region is the master biological clock of your central nervous system, the central metronome responsible for phase-locking every metabolic, hormonal, and cellular rhythm across the entire human body. The arrival of this photic signal at the suprachiasmatic nucleus initiates an immediate and profound metabolic reset across the entire human organism. Inside the 20,000 densely interconnected neurons of this master pacemaker, the surge of action potentials triggers a massive influx of intracellular calcium through voltage-gated channels and N-methyl D-aspartate receptors. This calcium wave activates mitogen-activated protein kinase cascades and phosphorylates the transcription factor CAMP response element binding protein. Within minutes, this activated protein binds to specific promoter sequences, driving the immediate transcription of core clock genes, particularly period 1, period 2, and cryptochrome. This rapid genomic activation abruptly recalibrates the central autoregulatory transcription-translation feedback loop, a molecular pendulum composed of interacting transcriptional activators and repressors that requires approximately 24 hours to execute a single cycle. Simultaneously, the suprachiasmatic nucleus transmits an inhibitory neural signal down an intricate polysynaptic circuit. Efferent projections descend from the hypothalamus to the intermediolateral cell column of the upper thoracic spinal cord, synapse onto preganglionic sympathetic neurons, and ascend through the sympathetic chain to the superior cervical ganglion. From there, postganglionic fibers innervate the pineal gland. Under daytime photic stimulation, this pathway sharply shuts off the release of norepinephrine. Deprived of adrenergic stimulation, the pinealocytes immediately deactivate arylalkylamine N-acetyltransferase, the primary rate-limiting enzyme required to convert serotonin into N-acetylserotonin and subsequently into melatonin. Melatonin synthesis halts abruptly. Circulating concentrations of the hormone collapse from their nighttime peak, clearing the bloodstream and removing the biological signal of subjective darkness. At that exact moment, the central pacemaker stimulates the paraventricular nucleus of the hypothalamus to initiate an opposing hormonal surge. Neurosecretory cells release corticotropin-releasing hormone into the hypophyseal portal system, prompting the anterior pituitary gland to discharge adrenocorticotropic hormone into systemic circulation. Upon reaching the zona fasciculata of the adrenal cortex, this hormone triggers the cortisol awakening response. Rather than a marker of chronic stress, this morning cortisol pulse is a finely tuned physiological ignition switch. Under its influence, core body temperature rises by approximately 0.5 degrees Fahrenheit. Peripheral vascular resistance increases slightly, activating systemic circulation. In the liver, glycogen phosphorylase is stimulated to break down stored glycogen, mobilizing a steady stream of glucose into the bloodstream to nourish the central nervous system and prime skeletal musculature for physical exertion. Neural alertness peaks, reaction times shorten, and cognitive processing speed accelerates. Yet the most profound mechanism set into motion by those morning photons is an invisible biochemical countdown. By abruptly suppressing pineal melatonin at dawn and phase-locking the electrical firing frequency of the suprachiasmatic nucleus to solar time, the morning photic pulse establishes the baseline calibration for an enzymatic timer. As the day progresses, extracellular adenosine steadily accumulates in the basal forebrain and cortex as a byproduct of cellular metabolism, generating homeostatic sleep pressure. But sleep pressure alone cannot induce restorative rest without circadian alignment. Approximately 12 to 14 hours after that initial morning light pulse, the suprachiasmatic firing rate naturally declines, releasing the inhibition on the pineal gland and permitting a clean, robust surge of endogenous melatonin. The onset, latency, continuity, and microarchitecture of your slow-wave delta sleep tonight are physically dictated by the precise nanometer wavelength of light that entered your pupils when you woke up this morning. Yet the eye is not merely a passive detector for calibrating the central neurological clock. It is an active mechanical organ whose physical geometric architecture is shaped directly by the ambient photon density of natural sunlight. If you examine the epidemiological data on human vision over the past 5 decades, you discover a global structural deformation occurring at a speed that completely defies genetic evolution. In the mid-20th century, roughly 20 to 30% of children and young adults in developed East Asian nations were myopic. Today, in urban centers across Singapore, South Korea, Taiwan, and China, the prevalence of myopia among adolescents has surged past 85%, with nearly 20% developing high myopia, a severe structural distortion that dramatically elevates the lifetime risk of retinal detachment, choroidal neovascularization, glaucoma, and irreversible visual loss. The United States and Western Europe are tracking the exact same upward trajectory, with myopia rates doubling over the identical time frame. To understand why this is happening, you have to look at the raw mechanics of ocular geometry. In an optically perfect human eye, a state known as emetropia, the refractive elements of the eye, specifically the curvature of the cornea and the pliable crystalline lens, bend incoming parallel rays of light so that they converge onto a singular, microscopically precise focal point on the central fovea of the retina. The physical distance between the anterior surface of the cornea and the posterior surface of the retina must match the focal length of those refractive lenses down to a fraction of a millimeter. When that distance is mismatched, vision fails. The mechanical pathology underlying the modern myopia epidemic is almost exclusively axial elongation. The physical eyeball stretches excessively from front to back, morphing from a spherical globe into an elongated prolate spheroid. A longitudinal elongation of just 1 single millimeter pushes the retina back far enough to cause a refractive error of nearly 3 diopters, forcing incoming light to focus entirely in front of the photoreceptors, rendering the distant physical world an unresolvable blur. For over a century, clinical dogma attributed this anatomical stretching to near-work strain. The working hypothesis suggested that prolonged reading of physical books, micro-focusing on small text, or staring at backlit digital screens caused chronic spasm of the ciliary muscles, physically distorting the scleral envelope. But in 2012, Professor Ian Morgan and an international team of researchers published a landmark systematic review and epidemiological investigation in The Lancet that overturned this model. Morgan showed that the sheer volume of near work, whether reading academic texts or staring at smartphone displays, is statistically decoupled from the onset of axial elongation when outdoor exposure is controlled for. The primary determinant of ocular stretching is not what children are looking at, but the photic environment in which they are looking at it. The structural trigger driving this global physical deformation is the chronic, severe deprivation of full-spectrum outdoor solar radiation during the critical developmental windows of ocular maturation. The human eye requires the raw photon flux of the open sun to govern its physical boundaries. The mechanism operating inside the ocular globe is governed entirely by photon flux density, measured in lux. To the human visual cortex, an office or school classroom illuminated by overhead fluorescent tubes or LED panels appears perfectly bright. The pupil dilates slightly, the neural circuitry adjusts its gain, and you perceive a well-lit space. But photometrically, indoor architecture is a biological void. Modern interior lighting rarely delivers more than 300 to 500 lux to the surface of the cornea. Step outside beneath a heavy cloud cover on an overcast morning, and the diffuse solar irradiance immediately floods the eye with 10,000 to 25,000 lux. Step onto an open street on a clear sunny afternoon, and that measurement surges past 100,000 lux, occasionally exceeding 130,000 lux at solar noon. The human visual system smooths over this massive 300-fold discrepancy, but the underlying cellular machinery of the eye measures every single photon. When light possessing this extreme outdoor photon density penetrates the clear media of the anterior chamber and strikes the neural retina, it engages a specialized population of retinal interneurons situated within the inner plexiform layer, dopaminergic amacrine cells. These cells do not participate in spatial resolution or image construction. Instead, they act as photic transducers. High-intensity illumination accelerates the enzymatic activity of tyrosine hydroxylase, the rate-limiting enzyme that converts the amino acid L-tyrosine into L-dopa, which is subsequently decarboxylated into dopamine. Under the sustained photon pressure of natural sunlight, these amacrine cells synthesize and exocytose substantial volumes of dopamine directly into the extracellular matrix, the vitreous humor, and the adjacent retinal pigment epithelium. This released dopamine operates as a biochemical brake on ocular growth. The neurotransmitter diffuses across the neural retina and the choroid to bind specifically with D2-like and D4 dopamine receptors expressed on the surface of scleral fibroblasts, the specialized structural cells responsible for maintaining the tough, fibrous outer coat of the eyeball. When dopamine occupies these receptors, it triggers an intracellular signaling pathway that inhibits the transcription and enzymatic activity of matrix metalloproteinase 2. Under normal conditions, the eye maintains a steady internal fluid pressure of 12 to 21 millimeters of mercury. To resist this hydrostatic outward force, the sclera requires a dense, highly cross-linked matrix of type 1 collagen fibrils and sulfated glycosaminoglycans. Dopamine signaling preserves this structural integrity, ordering the fibroblasts to maintain a rigid, highly tensile extracellular wall that arrests further longitudinal expansion. In the dim photic environment of indoor confinement, this signaling cascade collapses. Deprived of the threshold photon flux required to sustain tyrosine hydroxylase activation, retinal dopamine release drops by more than 80%. The biochemical stop signal vanishes. Without dopamine binding to its scleral receptors, local matrix metalloproteinases are unrestrained, actively digesting and remodeling the collagen framework. The sclera thins, loses its tensile stiffness, and yields to the eye's internal fluid pressure. The ocular globe stretches rearward millimeter by millimeter along its optical axis, turning a spherical optical instrument into an elongated prolate spheroid. Ian Morgan's data demonstrated that this structural deformation is entirely preventable. Supplying the developing visual system with 2 to 3 hours of ambient outdoor sunlight each day delivers the precise quantitative photon flux needed to sustain retinal dopamine tone, locking the physical architecture of the human eye into its correct geometric form. While the eye uses a fraction of a square inch of exposed retinal surface to set the master clock and mechanically tune the focal geometry of vision, the rest of your body presents an entirely different optical interface to the sky. Spread across approximately 20 square feet of surface area, your skin is the largest organ in your body, comprising roughly 16% of your total body mass. For over a century, classical dermatology and physiology treated this vast integumentary envelope as little more than a passive physical shield, a waterproof barrier engineered from dead, keratinized cells designed to keep pathogens out and internal moisture in, punctuated occasionally by the singular endocrine task of synthesizing vitamin D. Epidemiologists had long observed a physiological anomaly that classical biology could not explain. As early as the mid-20th century, large-scale population surveys revealed that resting systemic arterial blood pressure tracks an unmistakable latitudinal and seasonal gradient. The further you travel north or south from the equator, the higher the average blood pressure of the population climbs. Within any given latitude, cardiovascular mortality consistently peaks during the darkest months of midwinter and falls to its nadir during the height of midsummer. When researchers attempted to attribute this phenomenon entirely to vitamin D, clinical trials collapsed. Supplementing high-dose oral cholecalciferol to patients consistently failed to duplicate the systemic hypotensive protection observed in sun-exposed populations. Something else was happening directly inside the tissue under the influence of sunlight. In 2014, Dr. Richard Weller and an interdisciplinary team of photobiologists and clinical pharmacologists at the University of Edinburgh published a series of findings in the Journal of Investigative Dermatology that fundamentally redefined the cardiovascular role of human skin. Weller demonstrated that human skin is not merely an insulated boundary, but a massive light-activated chemical repository for nitrogen oxides. To understand the scale of this storage, consider the baseline biochemical disparity between the skin and the vascular bloodstream. Under normal resting conditions, circulating blood plasma contains relatively low concentrations of bioactive nitrogen intermediates, with free nitrite hovering between 50 and 200 nanomolar and nitrate around 20 to 40 micromolar. In the epidermis and the upper papillary dermis, Weller's team discovered immense pools of preformed, stable nitrogen oxide species. These stores exist as inorganic nitrite, inorganic nitrate, and a diverse family of S-nitrosylated proteins and low molecular weight S-nitrosothiols, such as S-nitrosoglutathione. When quantified per unit of tissue volume, the concentration of these photolabile nitrogen reserves within cutaneous tissue exceeds the concentration in circulating venous plasma by 10 to 30-fold. These nitrogen species do not originate from active transdermal gas exchange. They are deposited and concentrated into the extracellular matrix and within the cytoplasmic compartments of living keratinocytes and dermal fibroblasts over days and weeks as metabolic byproducts of systemic nitric oxide synthase activity and dietary intake. Held in chemical stasis within the collagenous architecture of the dermis, these molecules are thermodynamically stable at normal physiological temperatures. They remain locked within the tissue indefinitely without interacting with the systemic circulation, unless they are struck by photons of a specific quantum energy capable of breaking their covalent bonds. When sunlight containing ultraviolet A radiation, with wavelengths spanning 315 to 400 nanometers, strikes your skin, it behaves completely differently than the higher energy, shorter wavelength bands of the solar spectrum. Because UVA possesses longer wavelengths, it easily traverses the stratum corneum and the melanin-dense layers of the epidermis, penetrating directly into the vascularized papillary dermis to a depth of more than 1 millimeter. This is the precise anatomical zone where the microvascular network of the cutaneous circulation resides, woven directly through collagen fibers and dermal fibroblasts. Here, UVA photons encounter the vast reservoirs of preformed nitrogen oxides sitting accumulated within the extracellular matrix. What happens next bypasses classic enzymatic biology entirely and belongs strictly to quantum photochemistry. Most cellular signaling molecules require complex metabolic cascades, active transport mechanisms, specific enzyme availability, and substantial expenditures of cellular energy to be synthesized on demand. The cutaneous liberation of nitric oxide requires none of these biological intermediaries. It does not depend on the activity of endothelial nitric oxide synthase, and it consumes no cellular energy. It requires only the arrival of a single photon carrying the exact quantum energy necessary to shatter a covalent bond. Within S-nitrosothiol molecules, the sulfur-nitrogen covalent bond possesses a relatively low dissociation energy. A UVA photon in the 315 to 400 nanometer range carries an energy packet of approximately 3.1 to 3.9 electron volts. When this photon strikes the nitroso functional group, its electromagnetic energy is absorbed directly into the molecular orbital, triggering instantaneous homolytic cleavage of the sulfur-nitrogen bond. The chemical bond fractures immediately. This photolytic cleavage yields a stable sulfur intermediate and liberates free, active nitric oxide gas into the local tissue fluid, while simultaneously driving the photoreduction of adjacent nitrite reserves into additional bioactive nitric oxide. The liberated nitric oxide is an uncharged, nonpolar, lipophilic diatomic gas with a molecular weight of only 30 daltons. Because of its tiny physical profile and neutral charge, it encounters virtually zero resistance within human tissue. It does not require receptor-mediated transport, membrane pores, or vesicular packaging to move through the dermal environment. Instead, it diffuses radially outward in 3 dimensions at a velocity approaching 50 micrometers per second, moving with total physical freedom across aqueous interstitial fluid, cellular cytoplasm, and phospholipid cell membranes. Yet nitric oxide possesses a biological half-life measured in mere seconds before it is scavenged by circulating red blood cells or oxidized into inactive metabolites. This brief temporal window means that for the liberated gas to exert a meaningful physiological effect, its vascular target must be located within immediate physical proximity to the site of photon absorption. The microarchitecture of human skin is structured to exploit this exact physical constraint. The superficial dermal vascular plexus sits less than 200 micrometers beneath the epidermal boundary, forming a dense, high-surface area mesh of terminal arterioles, capillary loops, and postcapillary venules. Within milliseconds of UVA irradiation, the expanding wave of photoliberated nitric oxide gas traverses the perivascular space, crosses the single-cell endothelial lining of the dermal microvessels, and enters the cytoplasm of the underlying vascular smooth muscle cells that encircle the vessel walls. Once inside the vascular smooth muscle cell, nitric oxide immediately seeks out its primary biological target, soluble guanylyl cyclase. This enzyme contains a specialized heme prosthetic group centered around a ferrous iron atom. Nitric oxide binds to this iron core with extraordinary affinity, forming a 5-coordinate nitrosyl heme complex. This single sub-nanometer interaction forces a radical conformational shift across the entire enzymatic protein, accelerating its catalytic activity by more than 200-fold. Soluble guanylyl cyclase begins rapidly cleaving guanosine triphosphate, stripping away 2 phosphate groups to synthesize cyclic guanosine monophosphate, or cGMP. As intracellular concentrations of cGMP escalate, they engage and activate cyclic GMP-dependent protein kinase, commonly known as protein kinase G. This kinase acts as a master molecular switchboard for vascular tone. It swiftly phosphorylates several regulatory and transport proteins throughout the cell, orchestrating a rapid clearance of free calcium ions from the cytoplasm. Protein kinase G phosphorylates phospholamban, which relieves the inhibition on the sarcoplasmic reticulum calcium ATPase pump, allowing it to rapidly sequester cytosolic calcium back into the internal stores of the sarcoplasmic reticulum. Simultaneously, it stimulates large conductance calcium-activated potassium channels embedded in the plasma membrane. Potassium rushes outward, down its concentration gradient, driving the electrical potential across the membrane sharply negative. This state of hyperpolarization physically clamps shut the voltage-dependent L-type calcium channels, preventing any further extracellular calcium from entering. Without sufficient cytosolic calcium to bind calmodulin, myosin light chain kinase falls silent. Meanwhile, myosin light chain phosphatase remains active, stripping phosphate groups from the regulatory light chains of myosin. The cross-bridge interactions between actin and myosin filaments uncouple. The mechanical tension holding the smooth muscle taut dissolves. The microscopic muscle fibers encircling the vessel walls physically lengthen, allowing the luminal diameter of the dermal capillaries to expand dramatically. This local vascular relaxation is only the initial phase of a broader cardiovascular phenomenon. The human dermal microcirculation contains up to 10% of the body's total blood volume. As blood flows continuously through these irradiated, dilated capillary loops, the excess photoliberated nitric oxide binds to circulating plasma proteins and hemoglobin, forming stable circulating reservoirs of S-nitrosoalbumin and nitrite. These stable nitrogen carriers do not disintegrate in the skin. They are swept up into the venous return, passing through the cardiac chambers and into the systemic circulation, where they are propelled outward across the entire arterial tree. As these carriers transit through peripheral arterioles in the kidneys, the brain, and deep skeletal muscle beds, local metabolic conditions trigger the controlled re-release of active nitric oxide, inducing widespread arterial vasodilation. In their clinical trials at the University of Edinburgh, Dr. Richard Weller and his colleagues documented that a single physiological dose of UVA radiation produced an immediate statistically significant reduction in both systolic and diastolic blood pressure that persisted for hours after light exposure ceased. Weller verified that circulating concentrations of 25-hydroxyvitamin D remained completely unchanged throughout the observation period. The hypotensive response was not mediated by vitamin D. It was driven entirely by the photochemical mobilization of dermal nitrogen stores. Systemic hypertension remains the leading modifiable risk factor for stroke, myocardial infarction, and vascular dementia worldwide. Even a baseline reduction of 2 to 5 millimeters of mercury in systemic systolic pressure translates mathematically to a significant drop in long-term cardiovascular mortality. Through a purely non-enzymatic photon-driven reaction, sunlight functions as a direct transdermal vasodilator, actively reducing systemic peripheral resistance and lightening the mechanical workload imposed on the human heart with every beat. Yet the vascular liberation of nitric oxide represents only the first phase of the transdermal response to solar radiation. A completely distinct photochemical cascade begins across a shorter, higher-energy electromagnetic window, ultraviolet B radiation, occupying the narrow spectrum between 290 and 315 nanometers. While ultraviolet A photons penetrate deeply into the vascularized dermis, ultraviolet B photons carry higher quantum energy and are absorbed almost entirely within the stratified cellular architecture of the epidermis, specifically within the viable keratinocytes of the stratum basale and stratum spinosum. Embedded directly within the phospholipid bilayers of these epidermal cell membranes is a dense population of 7-dehydrocholesterol molecules, an immediate sterol intermediate produced during endogenous cholesterol biosynthesis. The 7-dehydrocholesterol molecule possesses a characteristic 4-ring cyclopentanoperhydrophenanthrene sterol backbone, distinguished by a conjugated diene system, 2 alternating double bonds located between carbons 5, 6, 7, and 8, within its second ring, the B ring. When a UVB photon within the 290 to 315 nanometer wavelength band penetrates the epidermis and collides with this conjugated diene, the energy of the photon matches the exact resonance frequency required to excite a pi electron from the highest occupied molecular orbital to the lowest unoccupied molecular orbital. This quantum absorption instantly destabilizes the thermodynamic equilibrium of the molecule, driving an electrocyclic 6-pi ring opening reaction. In accordance with the quantum mechanical rules governing pericyclic transformations, the covalent carbon-carbon single bond linking positions 9 and 10 of the steroid nucleus shears open. The cleavage of this single bond shatters the rigid integrity of the 4-ring sterol skeleton, converting 7-dehydrocholesterol into an unlocked flexible intermediate known as pre-vitamin D3, or pre-cholecalciferol. In its nascent state, pre-vitamin D3 exists in a sterically hindered cis conformation that is thermodynamically strained within the hydrophobic lipid bilayer of the cell membrane. What follows is an extraordinary manifestation of physical chemistry operating inside living tissue. The intermediate molecule requires no biological catalyst, no active transport, and no consumption of cellular adenosine triphosphate to complete its journey. Instead, driven exclusively by the ambient kinetic energy of mammalian body heat, maintained precisely at 98.6 degrees Fahrenheit, pre-vitamin D3 undergoes a slow, non-enzymatic thermal isomerization over the course of 24 to 48 hours. During this thermal equilibration, a hydrogen atom residing at carbon-9 shifts across the conjugated triene system to carbon-19 in a stereospecific 1,7-sigmatropic hydride rearrangement. This intramolecular shift relieves steric strain, rotating the orientation of the A-ring relative to the remaining ring fragments and yielding the thermodynamically stable corticosteroid known as cholecalciferol, or vitamin D3. This dermal photochemical factory contains an exquisite intrinsic regulatory mechanism that renders vitamin D toxicity from solar exposure physically impossible. If the skin remains exposed to continuous UVB irradiation, the rising concentration of pre-vitamin D3 does not simply produce runaway quantities of cholecalciferol. Because pre-vitamin D3 itself remains photolabile, additional incoming photons trigger secondary, reversible photoisomerization reactions, converting excess intermediate molecules into biologically inactive structural isomers, primarily lumisterol and tachysterol. If cholecalciferol itself begins to accumulate in the epidermal membranes before clearing into the blood, it absorbs excess ultraviolet radiation and decomposes into inert suprasterols. Through purely physical and photochemical kinetics, your skin acts as an autonomous pharmaceutical synthesizer that self-terminates production the moment saturation is reached, manufacturing precisely the therapeutic dosage required without the possibility of chemical overdose. Because cholecalciferol is a hydrophobic lipid, it cannot travel freely through the aqueous environment of the bloodstream. It is extruded through the cell membrane of the keratinocyte into the extracellular space, where it is captured with high affinity by a specialized transport glycoprotein, vitamin D-binding protein. Circulating through the dermal capillaries, this complex travels through the portal venous system to the liver. Inside hepatic hepatocytes, the enzyme cytochrome P452R1, a microsomal 25-hydroxylase, adds a hydroxyl group to carbon-25, synthesizing 25-hydroxyvitamin D, or calcidiol. Calcidiol is the primary circulating reservoir of vitamin D in your blood, the marker measured during routine clinical laboratory testing. But calcidiol remains biologically dormant until it reaches the proximal convoluted tubules of the kidneys, or the membranes of activated immune cells throughout peripheral tissues. In the renal cortex, the mitochondrial enzyme cytochrome P450-27B1, known as 1-alpha-hydroxylase, performs a second precise biochemical modification, attaching a hydroxyl group to carbon-1. This transforms the circulating precursor into 1025-dihydroxyvitamin D, or calcitriol. Calcitriol is not a conventional vitamin. It is a potent, system-wide corticosteroid hormone. Once synthesized or delivered locally into target tissues, calcitriol crosses cellular and nuclear membranes to bind with picomolar affinity to the intracellular vitamin D receptor, or VDR. Upon ligand binding, the VDR heterodimerizes with the retinoid X receptor, translocates directly to chromatin, and identifies specific promoter sequences known as vitamin D response elements across the human genome. Through this direct nuclear mechanism, sunlight modulates the transcriptional expression of more than 200 genes, with some genomic analyses identifying over 1,000 regulated sites, governing intestinal calcium absorption, systemic bone mineralization, and the transcription of cathelicidin and defensins, the primary antimicrobial peptides deployed by macrophages to destroy invading pathogens. Yet ultraviolet wavelengths represent only a fraction of the solar spectrum striking your body. Over 50% of the total radiant energy reaching the Earth from the Sun arrives in the near-infrared spectrum, spanning wavelengths from 650 to 1,000 nanometers. Unlike ultraviolet or visible light, near-infrared photons possess a unique optical penetration depth, passing cleanly through the epidermis, through subcutaneous adipose tissue, and deep into skeletal muscle and internal organs. In landmark work led by Professor Russell Reiter at the University of Texas Health Science Center, researchers uncovered an astonishing subcellular response to this invisible radiation. When near-infrared photons penetrate human cells, they are absorbed by cytochrome C oxidase, the terminal enzyme in the mitochondrial electron transport chain. This optical stimulation enhances cellular ATP synthesis and simultaneously upregulates the production of subcellular melatonin directly within the mitochondria. This mitochondrial melatonin never enters the bloodstream and has nothing to do with sleep cycles. It acts as an ultra-potent, localized antioxidant synthesized right at the primary source of oxidative stress, neutralizing reactive oxygen species, protecting mitochondrial DNA, and repairing cellular machinery in real time. And this photobiological dialogue extends directly into neurobiology. In 2014, researchers at Harvard Medical School, led by Dr. David Fisher, demonstrated that ultraviolet radiation striking epidermal keratinocytes activates the tumor suppressor protein p53, which rapidly upregulates transcription of the pro-opiomelanocortin, or POMC, gene. The resulting POMC prohormone is enzymatically cleaved not only into alpha-melanocyte-stimulating hormone to initiate protective pigmentation, but also into beta-endorphin, an endogenous opioid peptide. Released directly into the dermal microcirculation, beta-endorphin enters the systemic bloodstream and interacts with mu-opioid receptors throughout the nervous system, raising physical pain thresholds, elevating mood, and generating an evolutionarily conserved reward signal that encourages organisms to seek light. Sunlight is biochemically structured to reward you for stepping under it. When you synthesize all of these parallel mechanisms, circadian phase locking through retinal ganglion cells, systemic blood pressure reduction through dermal nitric oxide photolysis, genome-wide transcription through vitamin D synthesis, deep mitochondrial defense through near-infrared melatonin, and neurochemical elevation through beta-endorphin, the clinical significance becomes undeniable. In 2016, Professor Pelle Lindqvist and his colleagues published the results of a 20-year prospective cohort study involving 29,518 Swedish women in the Journal of Internal Medicine. The epidemiological data revealed that women with active sun exposure habits had a significantly lower risk of cardiovascular death and non-cancer mortality compared to those who avoided the sun. All-cause mortality was effectively doubled among habitual sun avoiders. Lindqvist concluded that complete avoidance of sun exposure represents a risk factor for overall mortality with a magnitude of impact comparable to chronic cigarette smoking. Sunlight is not passive ambient illumination. It is an unreplicable multi-wavelength pharmacological cocktail engineered by physics and evolutionary biology to regulate your vascular tone, your genetic expression, your cellular energetics, and your internal biological clock. Because ultraviolet radiation carries biological risks alongside its systemic benefits, understanding your individual skin phototype, monitoring ambient UV index, and consulting a qualified medical physician or dermatologist ensures you harness these mechanisms safely. Subscribe to Hidden Science for deep dives into the hidden biological mechanisms shaping human health and performance.